Lecture 3, Cells of Nervous System Neuronal Signaling. These slides are available to everyone in the class, and just reading it out loud to help me get ready for the next class, and also to familiarize myself with the concepts in the Lecture 3 slides so that if I get to ask one of the questions on one of the slides from the first lecture, to just research and think about brain topics, that I might be able to work in some of these concepts from these slides. Lecture 3, Cells of Nervous System Neuronal Signaling. Cellular organization of life. In 4th circa BCE, Democritus proclaimed that the physical structure of both animate and inanimate objects is built on blocks not visible to the naked eye. In the 17th circa CE, the development of microscopy confirmed that plants and animals are comprised of cells, the smallest unit of life. Embryological, morphological, and functional properties distinguish these cells in various tissues and organs, such as muscles, bones, skin, heart, etc. For the little graphic, make sure that you have the video playing at highest resolution, and most of the videos I try to have the setting available for 4K. So go and look in those settings and make sure you're watching the video in the highest resolution possible. Types of cells in the body: stem cells, bone cells, blood cells, muscle cells, fat cells, skin cells, and nerve cells is emphasized. It's circled. Endothelial cells, sex cells, pancreatic cells, pancreatic cells, cancer cells. Cells of the nervous system. Imaging all the cells in one small section of a mouse brain shows how densely packed the cells are in the brain. Brainbow, EM reconstruction. Cells of the nervous system. The nervous system is composed of two main types of cells. Neurons which process and communicate information, give rise to mental representations. Glial cells, which protect and nourish the neurons, play mostly supportive role. About equal amounts of glia cells and neurons in brain. Generic cell, myelin, neuron, glial cells. 5 types of glial cells. We have type, appearance, feature, and function. Ependymal cell, small, ovoid, secretes cerebrospinal fluid, CSF. Astrocyte, star-shaped, symmetrical, nutritive, and support function, blood-brain barrier. Microglial cell, small, mesodermally derived, defensive function, immune system. Oligodendroglial cell, oligodendrocyte, asymmetrical, forms myelin around axons in brain and spinal cord, myelin in CNS, central nervous system. Schwann cell, asymmetrical, wraps around peripheral nerves to form myelin. Myelin in PNS, which I think would mean peripheral nervous system. It's table 3.1. Neuron structure. Neurons come in all different shapes and sizes. Sensory neurons bring information to the central nervous system: dendrite, axon, bipolar neuron, retina, somatosensory neuron, skin, muscle. Wow. It's hard to read just because my screen's too small, even though it's relatively big. Interneurons associate sensory and motor activity in the central nervous system. Dendrites, axon, stellate cell, thalamus, pyramidal cell, cortex, Purkinje cell, cerebellum. Motor neurons send signals from the brain and spinal cord to muscles. Motor neurons send signals from the brain and spinal cord to muscles. Dendrites, axon, motor neuron, spinal cord. And some neurons are very long. It looks like a dinosaur. It's axon terminals, axon nerve cell body with nucleus. receptor terminals. Spines, dendritic protrusions. Spines, dendritic protrusions. Spines are located in dendrites. Neurons that contain spines are sometimes called spiny neurons. Spines are the site where excitatory signals are transmitted to cells, sites of synapses. Spine remodeling is thought to be important for learning and memory. It's a graphic, something like one. I don't know if that is that UM? Is it one? Does it stand for 1 nanometer? Dendrite, spine neck, spine head. And then the next graphic, dendrite, dendritic spines, synapse. Gray versus white matter. Gray matter refers to the cell bodies and dendrites of neurons. White matter refers to the axons. White matter refers to the axons. They are white because they are covered by myelin, a fatty substance. Where will most DNA be found? Transverse section of spinal cord, frontal plane through brain, transverse plane through spinal cord, gray matter, white matter, frontal section of brain. Signaling in the nervous system. Neuronal signaling involves electrical signals within and chemical signals between neurons. Dendrites, cell body, soma, nucleus, axon hillock, axon, direction of signal, axon terminals, presynaptic neuron, synapse, dendrites, axon terminal, neurotransmitter after release, postsynaptic neuron, axon terminals, collateral axon. Electron micrograph of a synapse. Synaptic vesicles, presynaptic neuron. Electrical and chemical signals in neurons, graded potentials. Nucleus, action potential, synaptic transmission. Dendrites collect electrical signals. Cell body integrates incoming signals and generates outgoing signal to axon. Axon passes electrical signals to dendrites of another cell or to an effector cell. Neurons and all other cells of the body maintain an electrical potential difference across their membrane. Extracellular space, intracellular fluid, lipid bilayer that forms neuronal membrane. The resting membrane potential of neurons is negative. Electrode enters neuron. Amplifier. Oscilloscope screen. Voltage, time at which electrode enters neuron. 0 MV could be millivolts, and I think it's at a resting membrane potential of minus 70 millivolts, and time. If I read any of this wrong, just let me know. Neurons are negatively charged inside when they are at rest. Neurons have ions, charged atoms, inside them and are also surrounded by fluid that has ions. Thus, a neuron has charges inside, and these are different from the ones outside, so there is a difference in electrical charge. We call this charge difference across the membrane potential. When neurons are not actively signaling, that is, at rest, their resting membrane potential is negative. Postsynaptic potentials, excitatory or inhibitory. Before transmitter release, presynaptic terminal. I don't know what V to the M means, that millivolts again. And minus 70 is the resting membrane potential, presynaptic neuron. Inhibitory postsynaptic potentials, IPSP. After transmitter release, presynaptic terminal, postsynaptic neuron. Did I just read that already? Oh no, it's different. So the first one was excitatory. Well, the first one was postsynaptic potentials. Then it was excitatory postsynaptic potentials. Then it's inhibitory postsynaptic potentials. I think I read this, right? After transmitter release, presynaptic terminal, resting membrane potential, postsynaptic neuron. Intracellular injection of currents to mimic an EPSP and its effect on membrane potential. EPSP is excitatory postsynaptic potentials. Excitatory postsynaptic potentials. So intracellular injection of current to mimic an EPST. Excitatory something something. Excitatory postsynaptic potentials. Excitatory postsynaptic potentials and its effect on membrane potential. Current step. We have 1, 2, 3, and then all were none amplitude, threshold, resting membrane potential, all were none amplitude, action potentials, spikes. Action potentials are the electrical signal in neurons. Action potentials initiate in the initial segment of the axon. If the membrane potential depolarizes above threshold, they always propagate down the axon towards the terminals. Action potential spikes are always the same amplitude anywhere in the axon. In other words, they are all or nothing. The properties of a stimulus are coded in the spiking rate of a neuron. Action potential, also known as spike, that's voltage in millivolts, I think. Action potential, threshold, time in milliseconds. Spiking activity of a neuron in the macaque parietal cortex. And it looks like there might even be a sound demonstration. Well, it doesn't work here, but maybe we'll hear it at lecture. So it's just a graph of voltage, millivolts, and time to time. And here's the oscilloscope. There's a anesthetized mouse, spiking activity in the dopamine center of the brain. Stimulation in cerebellum evokes short inhibition. Fast saltatory conduction. Fast saltatory conduction of spikes down the axon relies on myelin. There looks like Na+, K+. One of them's calcium, right? And what's Na+? Is it that stuff in bananas? The stuff that's good for you. I forgot what it is. Axon, Na+, K+, current flow, myelin node of Ranvier. Current flow, N plus K plus. Action potential jumps, saltare, rapidly from node to node. Oh, I wonder if Na plus is potassium. I knew it had something to do with the banana. Neurons communicate at synapses. Presynaptic neuron, postsynaptic neuron, axon hillock, dendritic spines. Electron micrograph of a synapse. Synapse. Electron micrograph of a synapse. Synaptic vesicles. Presynaptic neuron. Neurons have thousands of synaptic connections. Multiple synapses may signal at the same time onto this cell. Then responses add together, they summate, and you get synaptic integration. Synaptic integration. Dendrites in cell body have excitatory and inhibitory responses to neurotransmitters, EPSP, IPSP, cell body, axon hillock. If there are enough excitatory responses that get the axon initial segment to the threshold, then an action potential is generated. Initial segment, depolarized membrane at initial segment to threshold level, axon terminal generating an action potential, nerve impulse. Information is transmitted through synapses. Synaptic vesicles, small spheres in presynaptic axon terminals that contain a neurotransmitter, a specialized chemical. Neurotransmitters are released in response to spiking activity in the axon. Neurotransmitter receptors in the postsynaptic membrane are specialized proteins that react to neurotransmitter molecules. The postsynaptic receptors determine the action of the transmitter. The same transmitter may evoke EPSP or IPSP depending on the nature of the receptor on the postsynaptic membrane, an advantage of chemical transmission of info. Synaptic transmission, flow of information, presynaptic axon terminal, mitochondrion, synaptic vesicles, presynaptic membrane, neurotransmitter molecules, synaptic cleft, postsynaptic membrane, dendritic spine. When neurotransmitter molecules are released in the synaptic cleft, they bind to receptors on postsynaptic membrane and change the membrane potential of the postsynaptic neuron, electrical to chemical to electrical. Synapses are not static structures. New synapses can form and old synapses can be removed. Synaptic strength, how much neurotransmitter is released and or how many receptors are activated by it. Synaptic plasticity, dynamic changes in synaptic strength. Neurotransmitter, dendrite, I think that's potassium and calcium, but that one's like calcium 2+. Action potential, presynaptic terminal, spine, synaptic strength, more neurotransmitter, more receptors, new synapse, important mechanism for learning and memory. Neurons use neurotransmitters, NT, to communicate with each other. Neurotransmitters are small molecules released from the axon terminal of one cell onto another cell. Principal excitatory NT, which is neurotransmitter. Principal inhibitory neurotransmitter NT, brain. Principal inhibitory NT, spinal cord. So I see glutamate, GABA, and I don't know how to say that, especially with X through it. Modulatory NT, dopamine, serotonin, noradrenaline, acetylcholine, histamine. It says you don't need to know the structures of these molecules. That's good news. Four classical modulatory neurotransmitters. Acetylcholine, ACH, widespread loss of cholinergic neurons in brain is associated with Alzheimer's disease. ACH, acetylcholine, also does neuromuscular transmission targeted by venomous snakes. WMD. WMD sounds like weapons of mass destruction. In this context, Botox. Norepinephrine, NE, noradrenaline. Noradrenaline is important in alertness, mood, sexual behavior. Serotonin, 5-HT, controls mood, anxiety, sexual behavior, sleep, etc. Loss of 5-HT is prevalent. Theory for depression. Antidepressants like SSRIs enhance 5-HT. Dopamine, DA is important for motor control. Parkinson's disease results from loss of DA neurons. And for motivation can lead to addiction. Too much DA is a theory for schizophrenia. Antipsychotics suppress DA. Opiates, heroin, morphine, fentanyl, stimulants, methamphetamines, cocaine, and marijuana all enhance DA, dopamine, and are addictive. So after this, I will pull up one of the questions and see how much research I can do today. My feeling is I don't think I'll have an answer to the question I pull up, but we'll see. Because I still want to get to some memorization exercises too.